Hi everyone, good evening. We'll wait for 2-3 more minutes and then we'll start, okay?
Snehal, before you start, can you also click on record before you go?
Yeah, okay. Where is the record button?
You can go to tools or activities, either of those options, and you'll see record over there with transcripts.
Manage recording. Huh. Okay, let's begin. Okay, I was just reading the chat box. Yes, you should make notes during the class, of course. All right. Also,
This call is now taking notes.
okay. I will not be checking the text box while I'm teaching. After I'm done teaching, I'll open the chat box and then you can ask me your questions if there are any. Okay? Does that work? Because it keeps distracting me when I'm teaching. Okay? Let's go ahead. All right. Okay, let's start. So, just a minute. Okay, fine. So let's quickly have a recap of our day one. We talked about neurons and neuronal communication, right? So again, I'm just going to quickly walk you through this. So first we spoke about the parts of the neuron. We spoke about dendrite, cell body, nucleus, axon, myelin sheath, axon terminal, correct? Then we talked about ions. What are ions, right? So when our atom, when it loses electrons, it becomes a cation. It becomes positive. When it gains an electron, it becomes anion. So it becomes negative. Then we met our character, Nora the Explorer, and we spoke about how, when she's chilling, when our Nora is chilling, she is resting, right? So that is our resting membrane potential. And we spoke about how membrane potential is nothing but the voltage difference across the neuronal membrane, correct? We spoke about action potential. We spoke about how it is all or none electrical spike. We spoke about a few very, very important terminologies here. We spoke about depolarization, repolarization, hyperpolarization. So basically what happens is that when our neuron is chilling, when our Nora is chilling, right? And then suddenly it gets some message which is interesting enough. What is going to happen is that our Na plus channels, our sodium channels are going to open and our membrane is going to become more positive because it is going to let Na in, sodium in. Then we have repolarization. That is when our potassium ions are kicked out, right? So our membrane again comes back to negative state. And then there is something known as hyperpolarization, which is also known as the absolute refractory period, where the membrane becomes more negative, correct? And then it again comes back to our resting potential. So we had also briefly spoken about the difference between sodium potassium pump and sodium channel as well, where essentially we had said that sodium potassium pump is nothing but it is something that is always working, it is always active. Its only job is to push 3 sodium ions out and take 2 potassium ions in. Whereas when we talk about the sodium channel, it is a voltage gated channel. So what does it do? Its only job is to push Na into the neuron, right? Push sodium into the neuron. And it only opens very, very briefly during the depolarization phase, correct? And then I had shown you this graph. We had spoken about how at -70, Nora is chilling. And then it will go up, up, up. When depolarization happens, it reaches 0. And then when repolarization sort of happens, after reaching a peak at +30 action potential hoga, then it will come down and then hyperpolarization hoga, right? So basically that is what we had done so far. Yes? Are we clear with this part? Yes, no? All right. So now let's begin with our topic for today, which is structure of the nervous system. Okay. But before we begin, you know the drill. We are going to be starting with the MCQs just to check whether, just to check where we are at. Okay. And then we'll start from there. So the very first question, which of these correctly describes the central nervous system? What do you think? Correct. It is B. Perfect. Moving on. The tough outermost layer of the meninges is called yes, correct. It's A, dura mater. Next. Which of the following is the correct pathway of CSF flow through the ventricles? Now, this is challenging. Let's see how many of you get it right. What do you all think it is? Are we taking time to think? Okay. So the answer to this is C, but it's okay. This was a difficult question. We will get back to this later. I'll tell you what it is, why it is this way during the course of our lecture today. Okay, so pay attention. You think Palak thinks the order is different? It is C, lateral ventricle, third ventricle, cerebral aqueduct, and the fourth ventricle. We'll see how it goes, okay, in the lecture. Moving ahead. The cerebral cortex is part of which brain subdivision? What do you think it is? Correct. It's A, telencephalon. Very good. A lot of you are confusing it with diencephalon. That is not there. It's A, telencephalon. All right. Next. Which of the following is true about cerebrospinal fluid? What do you think it is? Okay. There's a lot of confusion in A and B. The right answer is B. It is produced by the choroid plexus. Again, we are going to cover all of these things today in the lecture, so don't worry. I'm sure by the end of the class, you'll know exactly what is it that we're talking about. Okay? Fine. So let's get started. Now I am not going to look at the text box. Okay? Fine. Just a minute. Okay. So last time we had followed Nora the neuron on her daily job, which is getting signals and processing them and sending them forward. But now Nora realizes, she looks around and she realizes, wait a second, I am not alone, right? She starts looking here and there and she sees thousands, millions of other neurons around her. She also realizes, yeah, some of them look like me, some of them, have longer axons, some of them have shorter axons. And now Nora gets curious. Now Nora starts saying, who am I? I mean, where am I? Who are my neighbors? What is this giant network that she's a part of? And then now imagine this, there is this wise old neuron who's, right next to her. And this wise old neuron tells her, Nora, you are not alone. You are a part of this huge thing called the central nervous system. And, we are the brain and the spinal cord. We are a part of it. And central nervous system is nothing but the headquarter of the whole body. And there's another neuron. Imagine another neuron shouting from further away, and who's saying that, listen, look here also. We are the part of the peripheral nervous system. Okay, and what do? We carry messages to and from the brain and the spinal cord, all the way to the muscles, the organs, the skin, right? And that is when it hits Nora that, okay, wow, I'm a part of a vast communication network. And so Nora is curious. Now Nora wants to know कि भाई ये है क्या? Because she is an explorer, right? So today we are going to explore along with Nora our nervous system. Okay. So let's start. What are the basic features of our nervous system? See, when you talk about our nervous system, it is primarily divided into two parts. Okay, first is our central nervous system, which includes our brain and the spinal cord. Okay, this is pretty basic. I think all of us know this. And second, we have something known as the peripheral nervous system, right? So it basically includes all the nerves which are outside CNS. So that is going to include your cranial nerves, your spinal nerves, right? It will also include peripheral ganglia. Okay, so this is basically what our nervous system is composed of. Okay. Now, before we even start with the explanation, I would first like to get you all acquainted with the directional terms. Okay. In the sense, जैसे हम map use करते हैं, there is north, south, east, west. Imagine Nora the explorer using such a map. Okay, but instead of north, south, east, west, we are going to be using different terminologies. So let's have a quick look at that. Although these terminologies may not be of so much of use for today's class, but for the future classes, I will be using those terminologies. So I need to familiarize you with those. Okay. So what are they? Now again, get ready for my Picasso drawing. Please do not laugh. Even if you're laughing, I won't get it. I mean, but still, don't. Okay, imagine this is a human being. Okay, this is our stick figure. Okay. Okay, I need to draw a bigger human being. Just a minute. Imagine this is our human being. Okay. Now, this is the brain here, and we have our spinal cord here, correct? Now, there are certain terminologies which I said regarding directions. So, what are these? When we are talking about the brain, okay, think of it in this way. This is dorsal, this is ventral, this is rostral, and this is caudal. Okay, what does this mean? Caudal basically means tail. Okay, caudal nothing is nothing but the posterior side. Okay, rostral basically means beak or nose. Okay, so this is, sorry, just a minute. Where did this go? Yeah. What is happening? Hmm. So this basically is anterior. Anterior. Okay. So when you're talking about the brain, when I say dorsal part, we're talking about something which is above. When I say ventral part of the brain, we're talking about something that is below. Okay. Rostral, beak, nose, something which is on your right hand side. Caudal meaning left hand side, which is back side of the brain, posterior side. Okay. Now, when I talk about the spinal cord, things are going to change a little. Okay. How is it going to change? Okay, think about it in this way. Our spinal cord is here, right? So our directions are going to change. Now, this is going to be rostral. This is going to be caudal. Excuse my handwriting. This is going to be ventral. And this is going to be dorsal. Okay. So here your dorsal is going to mean posterior. And your ventral is going to be anterior. All right. Are we clear with this part? So I would want you all to make a note of this in your notebooks, because now every now and then I will be using these terminologies itself. Okay, works? Fine. Again, I'm not looking at the chat box, so whatever questions you have, you can ask me later. Okay, now moving ahead. Fine. Now, when you talk about our brain, even before you start talking about your brain and the spinal cord, which is your central nervous system, there are certain protective structures, okay, around our brain and the spinal cord, which are known as the meninges. Now, it is very important for us to understand that these layers do not only cover the brain, but also the spinal cord. A lot of times when I've talked to students, I've realized that they think meninges are only for the brain, but that is not the case. They're also for the spinal cord. So what are these three protective layers? First, you have the dura mater. Okay. Second, you have the arachnoid matter. And third, you have the pia matter. Okay. Sorry, it's not double T, it's single T. Dura matter is basically the tough outer layer. Okay. Arachnoid matter is basically a spider web-like thing. Okay, it is the middle layer. And then you have the pia matter, which is very, very thin, which clings to brain's surface, like a very thin plastic sheet. Okay, very, very thin. In fact, if you remove the dura matter and the arachnoid matter, probably won't even be able to see what pia matter looks like, because it's that thin. Okay. Now, when there is inflammation of these meninges, something known as meningitis happens. So what is meningitis? It is, like I said, inflammation of the meninges. And if that happens, it can cause a lot of cognitive symptoms like confusion, and it can also affect consciousness. Okay. If you look at the diagram here, this is your dura mater. This part, the blue thing right here is your dura mater. The arachnoid matter is here. And this very thin thing is your pia mater. Yes. The same thing can be seen here in your spinal cord, right? There is dura mater, then in between is the arachnoid matter, and even inside of that is pia mater. Okay, are we clear with that? Fine. Now going ahead. Then there is something known as the ventricular system. Okay. First, even before we look at the slide, I'm sure this might seem a little overwhelming, even before we look at the slide. Okay. First, let's understand what are ventricles and why do we need them in the first place. See, ventricles by themselves are not doing things as such. Okay. Ventricles are nothing but these physical spaces that allow CSF, which is the cerebrospinal fluid, to even exist inside the space. Okay, what does the CSF do? It protects, it nourishes, and it cleans the brain. I'm sure all of us have learned about the CSF in our school, in our college, when we were studying basics of biopsych. So I'm not going to go too much into depth there. Okay? So without ventricles, our cerebrospinal fluid is not going to have any space to flow. And if it can't flow, it is if it ends up staying in one place, then the pressure is going to build up. And if that pressure builds up, it is going to lead to a lot of problems like hydrocephalus. What is hydrocephalus? Hydrocephalus is basically when there is a lot of fluid buildup, when there's a lot of CSF buildup, and that causes brain swelling. Okay, if ventricles are not there, then this will happen. Or if there is some problem with the ventricles, then this will happen. That is why ventricles are important, because they make it possible for the CSF to protect, to nourish, and clean the brain, right? So basically, there are four things that we need to keep in mind. There are four interconnected chambers. You have lateral ventricles, which you see right here. Then you have the third ventricle, which is right here. Then there is something known as the cerebral aqueduct here. If you see, it is connecting your third ventricle and the fourth ventricle with each other. And then you have the fourth ventricle. So basically what happens is that now you will think, okay, chalo, we have cerebral spinal fluid going right from the lateral ventricle to the fourth ventricle. But now what? So after the fourth ventricle, the cerebrospinal fluid is going to flow out into the subarachnoid space. Okay. Now subarachnoid space is going to be obviously around the brain and the spinal cord. And once the CSF flows out into the subarachnoid space, then it is going to get reabsorbed into the bloodstream. Okay. Are we clear with this part? So basically, what are the functions of CSF? It cushions the brain. It helps reduce weight. It also protects the brain from injury. Right? Now, where is CSF even produced in the first place? CSF is produced by something known as choroid plexus, something which was there in your MCQs. Okay. CSF is produced by the choroid plexus, which is there in your ventricles. Okay. So what is the flow going to be like? It is going to flow from the lateral ventricle to your third ventricle, then your cerebral aqueduct, then your fourth ventricle, then it will go around your brain. How will it go around your brain? Through the subarachnoid space. So I'm not going to write the entire thing here, you understand. And then it gets reabsorbed, okay, into the blood. Now, like I said, if there are certain CSF abnormalities, it might lead to hydrocephalus. Okay? And that is linked to a lot of cognitive impairments in memory and attention. Okay? Are we clear with this part? Right. Now, moving forward. Okay, before I move forward, till here, do we have any questions? Now I'm opening the chat box. Anything? Function of meningitis. Meningitis is something that happens when there is some kind of inflammation to your meninges. Okay. So it is reduce weight as in. So our brain is very heavy, right? So if it is inside CSF, the liquid is going to provide some kind of cushioning, which sort of makes it lighter. Okay. Reduces weight of the brain. Okay. Flow of CSF via diagram. Here, this diagram. Yes, CSF nourishes the brain. Yes. Okay. Is this diagram clear? Okay. Yeah, some of you are asking what is choroid plexus. See, choroid plexus is basically, think of it as a network of cells and blood vessels, and it is located inside the ventricles of the brain. तो उसका main function क्या है? To produce cerebrospinal fluid. ठीक है? It is basically, okay, I was not going to mention it because I thought it will be too much of jargon, but ठीक है. Choroid plexus is something which is made up of ependymal cells. ठीक है? What are ependymal cells now? These are specialized glial cells. Do you all know what are glial cells? We had not covered it the last time. But when we talk about neurons, we also talk about glial cells. Can you tell me quickly in chat box? Yes. Yes, they are supporting cells of the brain. Correct. Glial cells, also known as neuroglia, they are non-neuronal cells in the nervous system. जो क्या करते हैं? They protect and they nourish the neurons, right? So neurons, like our Nora, the explorer, what is she going to do? She is going to handle communication, right? So glial cells act like the support team. Okay, ensuring everything runs very, very smoothly. Those maybe there are different types of glial cells. We're not going to go into that today. But yeah, basically when you talk about choroid plexus, it is basically made up of ependymal cells, which are specialized glial cells. Okay, and it also has certain capillaries. ठीक है, और choroid plexus basically क्या करता है? एक अपना ना blood and cerebrospinal fluid का एक barrier होता है. ठीक है, so it plays, choroid plexus plays a role in maintaining that. So it basically helps to control the environment of CNS. Okay, are we clear with that? Where is CSF reabsorbed? In your bloodstream. Okay, some of you are asking what is subarachnoid space. So remember when I talked about meninges here, where did it go? Here. So subarachnoid space is basically the area between the arachnoid matter and the pia matter. So this is your arachnoid matter, and this very thin thing is your pia matter. Something which lies between that is your subarachnoid space. Okay, and that is basically filled with your cerebrospinal fluid. Okay, and it contains major blood vessels that supply the brain, and it also plays a role in shock absorption. Okay, and it also helps maintaining stable pressure in the brain. Okay, are we clear with that? Yes. Okay, great. Can I go ahead? All right. One person asked, why is CSF reabsorbed? Okay. Wait. What is this? Can you please explain directions again? I'll show you the slide. I'll just quickly explain bloodstream. Yes, where is CSF reabsorbed? It is reabsorbed in the bloodstream. Yes. This is the slide that I was talking about. If you all want to make a quick note or note of it, you can make that. The question is how come CSF in the spinal cord as well as absorbed into the blood? Okay. See, choroid plexus में CSF बनेगा, ठीक है? Now from the fourth ventricle, CFS is going to enter the subarachnoid space through very small openings. There are 3 small openings, ठीक है? Brain के ही end में, through which CSF enters the subarachnoid space. And in that way, it sort of bathes the brain and the spinal cord, which sort of helps providing in protection. Okay. Now, after CSF has moved to the subarachnoid space, then it goes into your bloodstream. Okay. Now, why does it go in the bloodstream? Think of it in this way. It, like I said, you know, CSF is important to build pressure. Now, cerebrospinal fluid and its drainage into the bloodstream is important to maintain homeostasis. Okay. If cerebrospinal fluid is not absorbed into the blood, then it is going to sort of build up and then it is going to increase intracranial pressure. Intra means inside the brain. So intracranial pressure, and then it will end up damaging the brain tissue. Okay? And body needs a way to remove old CSF while new CSF is being made. So the bloodstream sort of acts like a drainage system for your CSF. ठीक है? क्योंकि CSF क्या करता है? It carries waste products, it carries toxins away from the brain. Because apart from nourishing the brain, it also cleans up the brain, right? So once CSF enters the blood, these waste materials can be filtered out by organs like your liver, your kidney, and then they get removed from the body. Are you getting this? Is this clear? Bloodstream कहाँ जाएगा मतलब? Bloodstream, I mean, blood is there in your entire body, isn't it? Okay. So this blood, this waste materials are going to be filtered by organs like liver and kidney. Fine. All right. Now I'm closing the chat box and now we are going to go ahead. Okay. Fine. Now let's go ahead. Closing the chat box. All right. Okay. Now, development of the CNS. I purposely put this diagram here because a lot of times we get confused between the words like embryo and embryonic stage, fetal stage. So embryonic stage comes first, then comes the fetal stage. So if you look at the diagram right here, 3 to 8 weeks you already see forebrain, midbrain, hindbrain sort of starting to develop, right? Now, what exactly happens is that at embryonic stage, which is this stage, between 3 to 8 weeks, what happens is something known as neurogenesis. ठीक है? अभी neurogenesis क्या होता है? We spoke about Nora the Explorer, right? But how did Nora even exist in the first place? So for Nora to exist, we first had something known as neural stem cells. ठीक है? ये neural stem cells have the potential to generate all different kinds of neural cell types. ठीक है? तो ये stem cells ना differentiate into 2 types of cells. Neuronal progenitor cells and glial progenitor cells. So these neuronal progenitor cells are going to give rise to neurons. I mean, as obvious from the name. And the glial progenitor cells are going to give rise to astrocyte and oligodendrocytes. Okay. This entire thing is known as gliogenesis and neurogenesis. Again, very important for us to remember. Okay, developmentally. Now, there is something known as the neural tube. Okay. Now, by day 28 post-conception, a neural tube is going to get formed. Okay. And this neural tube eventually gives rise to your brain and your spinal cord. Okay. Now, how does it look like? Imagine like this is your neural tube. Okay. Now, some part of it is going to develop into brain. Some part of it is going to develop into spinal cord. Okay. This entire thing is your neural tube. Now, this is going to be rostral. This is going to be caudal, obviously, because caudal means tail. Correct? Now, surrounding this, okay, surrounding this neural tube is a very, very thick sort of a thing, which is called neural crest. So what does this neural crest do? This is something which is eventually going to give rise to most of your PNS. Okay, so your neural tube gives rise to brain and spinal cord, and your neural crest gives rise to your most of your peripheral nervous system. Okay. Now, also you need to remember that by the 7th month of pregnancy, the fetus will now start to emit its own brain waves. Pretty cool, ain't it? So, this is the time when you will see new neurons getting formed. You'll also see new synapses getting formed. You know what a synapse is, right? When there are two neurons, the gap between them, right, is synapse. So new neurons are getting formed, new synapses are getting formed by the brain at an extremely high rate. ये 7th month of pregnancy के time पे, ठीक है? And we also need to remember that even after the baby is born, the development doesn't stop at birth. In fact, most of the neurons, since we were just talking about neurogenesis, most of the neurons, are born during the first several years of your human life. Okay. In fact, till one is like 2 or 3 years old, axons are not fully myelinated also. Okay. And when we talk about synaptic connections which are formed, a lot of it peaks within the first year of your life, and then it starts steadily declining. Okay. Now, ये जो decline होता है ना synapses का, मतलब let's say there are neurons and now they are like forming connections with each other. There are a lot of synaptic connections being built within the first year of life. Eventually, our brain starts realizing that, okay, not all of these connections are important. We can let go of certain connections. Now that is called synaptic pruning. Okay. What is synaptic pruning? Synaptic pruning is basically brain's way of removing connections in the brain that are no longer required. ठीक है? तो ये synaptic pruning चालू रहता है until your teenage years. ठीक है? In fact, you will learn about this in abnormal psychology, I think. When we talk about autism spectrum disorder, it has been said that in early stages, there's excessive early brain growth, and that sort of leads to reduced pruning. The brain doesn't realize that some connections are not required. So that leads to hyperconnectivity, and that eventually leads to deficits in social and cognitive processing. Conversely, there is something known as childhood onset schizophrenia. Okay. Now it has been said that in childhood onset schizophrenia, excessive synaptic pruning होता है, जिसकी वजह से gray matter. We'll learn what gray matter and white matter is later on, but just remember this. Research has said that because of excessive synaptic pruning, gray matter का थोड़ा लॉस होना स्टार्ट होता है एंड कॉग्निटिव डिक्लाइन होना स्टार्ट होता है. सो इफ यू सी ऑटिज्म स्पेक्ट्रम डिसऑर्डर में रिड्यूस्ड प्रूनिंग है, दैट इज व्हाई देयर इज एन इशू, एंड इन चाइल्डहुड ऑनसेट स्किज़ोफ्रेनिया एक्सेसिव साइनेप्टिक प्रूनिंग है, दैट इज व्हाई देयर इज एन इशू. ठीक है, fine, but that is all of this is developmental stuff which I wanted you to remember. All right, this is the white matter and gray matter thing that I was talking about. Remember we had spoken about Nora, correct? So we had also spoken about myelin sheath. So myelin क्या करता है? Myelin sort of lends a white appearance, ठीक है, to regions where axons are a lot, like in number. ठीक है. So on the contrary, cell dense regions, ठीक है, जहां पे अपना सोमा होगा, they are going to appear gray, and places where there are a lot of axons, they are going to appear white. ठीक है. So gray matter is basically where all our cell dense regions exist. So this entire thing, these are cell dense regions, right? And white matter is basically the area where you are going to be seeing a lot of myelinated axons. Okay? So if you notice this entire thing, the dark thing, this is basically your cerebral cortex. So the outer gray part of the cerebral cortex, like I said, is where the gray matter exists, and the inner part, jahan pe axons hoga, that is going to be your white matter. Okay, and these are your ventricles. Okay, now heads up, now we're going to be looking at a diagram which may look very complicated but isn't. Okay, no need to get overwhelmed. So these are basic embryological subdivisions of the human brain. Okay, you see here, again, you don't need to remember all of this. I'll just put this because we were talking about development of the brain. So I thought this will be a little helpful for us to understand. Okay, so developmentally speaking, by the end of the first month of gestation period, kya hoga? The neural tube which I had drawn, now that is going to start closing. Okay, I had drawn that this beautiful Picasso drawing that I had done of the neural tube, where we spoke about one part being brain and one part being your spinal cord. So by the first month of gestation, by the end of it, the neural tube is going to start closing and 3 swellings are going to appear. Okay? And these 3 swellings are going to appear at, like if you see here, like this. Okay? So you will see prosencephalon, mesencephalon, rhombencephalon. Okay? Prosencephalon means what? Pro, forward, front brain. Okay? Now, eventually, this prosencephalon is going to get divided into 2 parts, which is telencephalon and diencephalon. Okay? Telencephalon, telen means end brain or outer brain. Telencephalon ka kya banega? Cerebral hemispheres. All of this we are going to look absolutely in detail in the next slide, so don't worry about it. Okay? Then hoga diencephalon. Diencephalon means, encephalon means brain. Diencephalon means through the brain or between the brain. So you will see thalamus and hypothalamus forming here. Okay? And all of these structures together are going to form your forebrain. Okay? Yeah. And then you have the mesencephalon. Mesencephalon is basically this middle swelling right here. Okay? it does not divide further. It stays intact and it eventually becomes your midbrain in adulthood. Okay. And the space which is going to be inside the midbrain is going to be your cerebral aqueduct. Okay. And then you have your rhombencephalon. Okay. Fine. Like I said, this is just for you to get some context. We are going to look at each and every part in some detail in the following lectures. Okay. So this is the table that we will be coming back to every now and then. Today, because we don't have so much time left, we're only going to focus on cerebral cortex and cerebral nuclei. Okay, that also we are not going to go in too much detail. We'll have another session dedicated to cerebral cortex and the four lobes. Right now, I'm only giving you a basic overview of what our CNS looks like. Okay? So this table is very important. I would want you all to, make a note of this in your notebooks, because after I finish every part, we'll come back to this table, we'll revise and we'll go ahead. Okay. Now, when you talk about the telencephalon, which is here, this part, okay, which is जो हमने prosencephalon देखा था in that neural tube का part, which then I said divides into two parts, telencephalon and diencephalon. So first we are going to look at the telencephalon. The telencephalon has two main parts, your cerebral cortex and cerebral nuclei. Cerebral cortex is nothing but basically your four lobes. Okay, if I want to really simplify it. Okay. Now again, time for some Picasso drawing. Now imagine this is your brain. Okay. This is your brain. I'm telling you that. Okay. And now it is like this. There are two hemispheres, right? You have the left hemisphere and the right hemisphere. Correct? This is basically your lateral view. Okay. So usually lateral view मतलब if you're only looking at it from a side, तो तुमको at any point of time सिर्फ एक ही hemisphere दिखेगा. You will not see both the hemispheres, correct? You're not going to see this part. If we take a lateral view, you're only going to see one hemisphere. Okay. So taking the lateral view, this is what the brain would look like. So the cerebral cortex essentially has four lobes: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. Now, there is something known as gyri and sulci. Okay, which all should remember. What is gyri? Or gyri or gyri, however you want to pronounce it. Some people call it gyri. There is gyri and there's sulci. Now the way I like to remember is sulci means sulking. So it is something that goes down. Gyri means something which is going upper. So gyri are the bumps and sulci are the grooves. Okay. So when you talk about the cerebral cortex, it is the outermost layer of the brain, and cortex literally means bark in Latin, bark of a tree. Okay, because bark of the tree is also dark in color. It is thin, but it is very tough, right? Our cerebral cortex is also like that. It is full of gray matter. It is tough. It is very thin. Now, our entire the outermost layer of this brain is folded into gyri and sulcis. And why is that? Because there's a lot of like bigger surface area, right? That needs to be fit inside the skull. So for that, we have these folds. Okay. Now, the telencephalon gives rise to most of the cerebral hemispheres. Okay. So this is where your four lobes come into picture. Now, there is something which divides your frontal lobe and the parietal lobe, like this part right here. Okay. That is called your central sulcus. Okay. Now, there is something that divides your parietal lobe from your occipital lobe. This part right here is called your parieto-occipital sulcus. Now, right here, where your occipital lobe ends and your temporal lobe sort of begins, there is something known as your pre-occipital notch. Okay? And there is something that sort of makes a demarcation between your frontal lobe and your temporal lobe. Right here. That is your lateral fissure. Okay, are we clear with this part? Fine. So cerebral cortex is clear. So this part is done. Okay. Now frontal lobes, like I said, we are going to be talking about the cerebral cortex and the four lobes much more in detail. So don't worry about that. But to quickly check, if you all, now I'm opening the chat box. If you all could quickly tell me, what does frontal lobe do? What does parietal lobe do? What does temporal lobe do? It will be great. Yeah, I'll show you the last two slides. Which one? This one? Okay. Yes. What do the lobes do was my question. Like just in one line, if you could tell me. Parietal touch, temperature, pain, yes. Occipital vision, yes. Temporal audition. Okay. Correct. You all are correct here. When you talk about frontal lobe, we're basically looking at planning, decision making, problem solving. There is something known as Broca's motor speech area, which also lies in your frontal lobe. Then a lot of your personality, impulse control, is implied in frontal lobe and the prefrontal cortex area. Then your parietal lobe, like you all correctly mentioned, it has to do with your sensory perception. So touch, pain, temperature. It has to do with your spatial awareness, your body orientation, right? Your somatosensory cortex is here. Then you have your temporal lobe, which basically involves hearing, language comprehension. Your Wernicke's area is here. Then there is memory formation. Then there is emotion processing. Uh, that is there in temporal lobe. Then you have your occipital lobe, which is basically very important for vision and visual interpretation. So color, shape, distance, depth, all of these things. Okay. So, if you want a quick mnemonic, again, this is very, very simplified, but just in case you want. When you talk about frontal lobe, okay, we'll look at F. It has to do with focus and function. When you talk about your parietal lobe, P, it has to do with perception or like proprioception. Okay, or position. When you talk about temporal lobe, sorry, what just happened? Just a minute. Yeah, temporal lobe. So T, we can call it talk and tune. Okay, talk in the sense not like motor speech area, like more of comprehension. I'm just trying to, make a mnemonic. And then you have occipital area, occipital lobe. Okay, O. So optics. Does this make it a little easier for us to remember? Yes, no? Yeah? All right. Fine. Okay. Now we're running out of time. So I'll quickly talk about the basal ganglia, the amygdala, and the basal forebrain. Very quickly, it will not take so much time. Okay? Fine. Now moving ahead with the basal ganglia part. Now understand, let's orient ourselves. We're done with talking about cerebral cortex. Now we're talking about the cerebral nuclei. So first we're talking about the basal ganglia. So what is basal ganglia? It is, just a minute. Yeah, it is involved in motor control. So it is going to speak about starting, stopping, regulating your voluntary movements. ठीक है, तो बेसिल गैंग्लिया बेसिकली क्या करता है? दिस इज द प्लेस वेयर ऑल योर मूवमेंट्स आर ऑर्गेनाइज्ड एंड दे आर स्मूथेंड आउट. सो थिंक ऑफ इट लाइक एज अ ब्रेन्स मूवमेंट ट्रैफिक कंट्रोलर. ठीक है, so it is going to allow desired movements to happen smoothly and is going to stop the unwanted ones. ठीक है, so it basically initiates all your voluntary movements. So it is also involved in motor learning. So all your habit formation or the skills that you sort of develop, this is very much implied there. ठीक है, it also contributes to action selection. मतलब which motor plan to execute in the first place. So like I said, traffic controller जैसा है, green and red light system जैसा है. तो if it tells you okay go, if it gives you a green signal, it is going to allow the intended action. If it shows you the red signal, if it says stop, then it is going to block the unintended movement. ठीक है. In fact, you must have heard about basal ganglia when you hear of disorders like Parkinson's, right? क्योंकि Parkinson में क्या होता है? The dopamine input, where we've not spoken about the neurotransmitters yet, but just hear me out. So dopamine input जो है, if it reduces, if it decreases to the basal ganglia, तो Parkinson's disease हमें दिखता है, जहाँ पे there is difficulty starting movement, there are slow movements, there is muscle rigidity, there is tremors, right? And opposite, अगर overactive basal ganglia है, so you will see a lot of repetitive behaviors. Now, when I say repetitive behaviors, what does it remind you of? What disorder does it remind you of? Can anyone tell me? Correct. OCD. Perfect. Yes. If I talk about, I said it has to do with a lot of intended movements, right? Now, if there is some issue and if there are a lot of involuntary movements or if there are involuntary sounds, what disorder does it remind you of? If I say involuntary movement or involuntary sounds, if I say ticks, yes, correct, Tourette's, beautiful, very good, correct. And yes, some of you said Huntington's, that is also correct. If basal ganglia circuits are overactive, तो क्या होगा? Huntington's होगा, involuntary jerky movements दिखेंगे. ठीक है? Then we talk about amygdala. Now amygdala is this small almond-shaped thing. ठीक है? Which lies in your medial temporal lobe, and it literally means almond in Greek. ठीक है? Now it is best known for its fear and emotion center. I know it's 7. I'll just wrap it up in the next 5-6 minutes. Okay. So yeah, it is your fear and emotion center. This is the place where amygdala processes emotions. It looks at fear, anger, threat detection. In fact, it also helps form emotional memories, especially the fear-based ones. Okay, and it is also going to play a role in recognizing the emotional expressions in other people's faces, right? And it also links emotions to your physiological responses, like your increased heart rate when you're scared. We haven't spoken about hippocampus yet, but just we'll talk about it later. But at least one thing we know for sure is hippocampus has to do something with memory, correct? So think of it in this way: if hippocampus stores facts, then amygdala stores feelings which are tied to those facts. ठीक है? तो amygdala बहुत लोगों से बात करता है, आजू बाजू के लोगों से. It interacts with hypothalamus, it interacts with hippocampus, it interacts with prefrontal cortex. ठीक है? तो anxiety disorders में क्या होगा? There is going to be heightened amygdala activation. ठीक है? Even when there is non-threatening stimuli. ठीक है? Because our body sometimes can't differentiate between what is a threat and what is a perceived threat. So anxiety disorders में you will see a lot of amygdala activation. Fine. This is just for you to get some context. This is your amygdala, this is your thalamus, this is your hippocampus, this blue region. Cool? And then we have your basal forebrain. This is the last part. ठीक है? Basal forebrain क्या करेगा? Basal forebrain is basically a group of structures which are involved in cholinergic projections to the cortex and hippocampus. Cholinergic means what? There is something, there is a neurotransmitter known as acetylcholine. ठीक है, ये एक chemical messenger होता है. What does it do? It helps the nerve cells talk to each other. So it is involved in basically three basic jobs है उसके, ठीक है. First, acetylcholine क्या करता है? It helps you move your muscles. ठीक है. Without acetylcholine, your brain can't tell your muscles to move in the first place. So for example, every time you wave your hand, okay, how is it making it happen? It is making it happen because acetylcholine is existing. Okay. Second, it helps you pay attention. It helps you stay alert. Okay. It acetylcholine tells your brain कि भाई अभी उठो, you have to focus, right? And another thing that acetylcholine helps us do is it helps you form new memories. Okay, and it helps you learn things. It helps the brain store new information. Okay, so agar there is degeneration of basal forebrain cholinergic neurons, then it is going to be linked to memory impairment. And if that happens, then what disorder is it? Is it giving? Like what disorder do you think is implied? Yes, Alzheimer's. Correct. Perfect. Very good. Very good. Fine. So, are we clear with this part? So, now let's quickly sum up. Okay, we'll take the MCQs first. And then once we finish all of that, then we can take the questions if we still have time, or we can take the questions next time. All right, because because you have other class also. Fine. We'll skip this question. I didn't do this. The subarachnoid space lies between which two meninges? Can anyone tell me what is it? People, can you all respond? The subarachnoid space lies between which two meninges? Am I audible? Okay, okay. So batao, where does it lie? We discussed this, isn't it? So can you all tell me what is the answer? Ari, why are you just sending thumbs up? Oh, I'm not able to see it. I'm just seeing up. I did. Okay, okay. Now I can see it. I can see it now. Yes. Okay, okay. Thank you. Yes. Yes, B. It is arachnoid membrane and pia matter. Perfect. All right. Moving ahead. Which of the following is not a function of cerebrospinal fluid? What do you think it is? Yes, correct. It is producing neurotransmitters to the cortex, obviously. I mean, this is an easy one. Next, damage to which lobe would most likely result in difficulty planning, making decisions, and controlling impulses? Yes, correct. Frontal lobe. Beautiful. Next, which of the following is the correct pathway of cerebrospinal fluid through the ventricles? Now, this is the same question, guys. Come on. We know this. Perfect. It is lateral ventricles, third ventricle, cerebral aqueduct, and fourth ventricle. Beautiful. And yeah, that's it. All right. Now, for the next 2 minutes, I'll answer your questions if you have any, because I really don't want to take up the next lecturer's time. Batao. Why are we using rostral? Bhai, ye map jaisa hai. Like north, south, east, west. These are pretty standard things that we usually follow. That is what you will see. How to not forget the names. Which names? This rostral caudal. I think one way for neuropsychology at least. I'll show the neural stem cells. Which you want to, are you hinting towards my Picasso drawing, Jennifer? The neural stem? Which one do you want? This one? The chart, this one. Acha, acha, acha. Sorry. This. Got it, yes. Sorry, I was answering your question. Khushi, how to not forget names. I think for neuropsychology, the only way is to keep reading it again and again and to say it out loud. Okay, when I take neuropsych offline also, I ask my students, every time I teach something, I ask people to repeat it after me because that is the only way you'll get it into your head. Yes, Palak, what acetylcholine doubt did you have? You want to know what acetylcholine is? Some of you are asking, don't you think neuropsych is heavy? It is biology. No, it is going to have a lot of complex terms to remember. But I think once, like I said, you keep familiarizing yourself with all the terms. And at least with my classes, before I move on to the next part, I will revise what we have done previously. So I think that will help. And yeah, any recommendations? Okay. So there are many books. There is something known as Zilmer. Then there is Kolb, Kolb, Kolb. There is Kalat, Kalat. There is Pinel, yes. All right. What can't you visualize? I mean, you have diagrams on the internet and textbooks are pretty good. All of them have diagrams in it. So you can always look at that. Okay. Yes, these are the 4 books. All right, fine. I think we don't have any more questions. Or do we have any more? Should we wrap it up then? All right, fine. By the way, for someone who had acetylcholine cut out, it is just a neurotransmitter. It is going to help in muscle contraction. It is going to help in learning and memory and stuff like that. We'll talk about neurotransmitters later, not now. Okay? All right, fine. I hope you all understood everything. And yeah, okay. See you later. Bye-bye. Shannon, over to you.
Hello, people. Snail, you can also stop recording. पहले शेयर करना बंद कर दो. Hello, people. Your brain is kind of fried. Isn't everyone's? Hey, UGC has extended the date. Why are they like this? Why was I up till 10:30 trying to navigate that shitty website, which kept crashing every 3 seconds? They not like us. It's extended to 12th. 12th, right? Have they? Yes, they've extended to 12th. सब ठीक हो जाएगा एक दिन. I like the optimism, but the evidence for that is not really up to mark. The count on the board keeps going down. That is how a counter works. As days go by, the count decreases. Exam dates bhi kara do. Wait, have they revealed the dates? Yeah, not end of June, usually mid June रहता है. Oh, last week, eh? Usually it's second, third week. Yeah. Okay, maybe it might. It says on the notification 21st to 31st June. It's saying that. मुझे नहीं दिखा. Okay, I might be blind. Never mind. ठीक है, no problem. More time to do this. ठीक है. I know your brains might be fried. By June session, June में ही लेना पड़ेगा. No, they'll call you for a retest. Don't worry. You give the exam twice. You know, the first is like a trial round, like a practice mock round. They are giving you mock test. That's so sweet. They're like, you come to this location, you travel like 3 hours, you give mock test and go. It's so sweet. You know, no other government, no other country cares this much for the students that they are providing mock test. Pragati. Cephalocaudal is like the top. Net and sweet in one sentence. MPhil finals during that time. Damn. Two exams to prepare for now. Okay, go. Take a quick break. 2-minute break, drink water, everything, and come back. Okay. And then we'll start with stats. Yeah. Humans and Phil and MA final exam on the same date. Damn. Yeah, they didn't confirm it. Wait, no, it's 24th or 25th. It can be either day. They've not confirmed it. Usually it's the first day though. Usually it ends up being the first day. Yeah. For 25th is a Sunday, no? So it's better if it's on 25th. One more day extra also. Usually they reserve the PhD thing for Sunday because they end up, people who apply that end up being usually are older than the MPhil crowd. So if they have to take work, say chutti or something, it's easier. Okay, let's start. So what have we done up until now? What do you remember? What do you know? Oh, before I start, I have to tell someone to record. Oh, it's still recording. Oh, it's not. Sanjana, would you be able to? Or because you're going live, it would not, it would have put a strain on your thing. Okay. Snehal, are you staying here for the rest of the lecture?
No, Ray, I was just looking at the chats. I'll leave now. Bye
No,
bye.
because I've asked you to record.
No, I can, but I'm not going to stay here the entire time.
Okay,
You ask
no.
Sanjana to record. Okay. Bye bye.
Bye bye. Anyone else interested? Arya, would you be, do you have stable net? Won't take much. You're here, Sarah? Acha. Till 8. Okay. Then I have to wrap up by it. Okay. No problem. Where is Sarah on the chat? Or is there any other Arya? But take it. We'll give it to Sarah and have to wrap up in 40 minutes. Take it. Yes, till she sets that up. I saw some stuff. Deviance by, variance by. Hey, variance gangster. Don't just give call ever. Deviance, variance, standard deviation. Take it. Let me open my thing. And I'm joining from another device as well so that I can also see the chats. Awesome. Let's start. Okay. What we have looked at so far is all of this, right? We've done all of this. We've looked at deviance by, what his job is, what he does. Deviance by is x minus m. The fact that he's weak by himself, that deviance will always, the deviations, the sum of deviations will always equal 0, right? This is the sum of deviations, right? This is the sum. Oh, wait, I'll put the pencil. The sum will always be 0. Sum of deviance will always be 0. So to make up for it, he borrows ammunition, which is why he becomes squared, right? And that results, that comes from variance gangster. This makes him variance, which is going to be very important throughout parametric psych. This one formula, that is your summation of deviance, right? X minus m, which is then squared so that it doesn't become, 0. And the average of that is n. This one thing is very important throughout all, which be it your correlation, be it covariance. We'll take a brief look at covariance today. And ANOVA, t-test, whatever it is, it keeps coming up again and again, or at least deviance keeps coming again and again. The deviation squared, sum of square deviations, it keeps coming again and again. Okay, we've looked at all of this, so I won't spend much time. Standard deviation, TCAP. So today we're looking at a new character. The new character is none other than standard error boy. Standard error, we'll call him SC. The standard error boy. Standard error is here to judge this guy. So standard deviation, what does the standard deviation do? So far you've learned it, right? We've been going over that. What is standard? What is this accomplishing? We went through all this to get deviance by, to make him squared, classify. Okay, let's put it more formally then. What is it? What is deviance by really doing? What is standard deviation really doing? Average of deviance, average of sum of squared deviations. Let's call it that for now, right? Because so far we've been calling it deviance, but let's call it properly average of the sum of squared deviations. This is just put in D for now, but this is deviance by and squared, right? The average of that. But what is the point of it? The point of it is very simply to tell you how noisy your data is. Like even if I'm classifying, if there's too many things on the either side of the mean, it means there's too much noise. It's too noisy then. And that is what it's telling you. It's more like an assessment of the mean, right? Let's say I have one sample over here and another sample over here. I've taken a sample, each are of 100, a sample of 100. This has a mean of 10. This has a mean of 10. This has a, okay, I'll write M over here and I'll write HT over here. If this has a standard deviation of 2 and this has a standard deviation of 20, which mean is more trustworthy in a way? Like which mean is a better representative? HD21, right? Because this is less noisy. It's less noisy than this guy. And that is what it's telling you. Yeah, the first one. Because this is less noisy. 2 as in the 2 one, the one which has the first one, yes, the one that has HD of 2. What would the graphs look like? The graphs would very simply be like if I draw it on a normal curve, and if it's 10, again, I don't really, it would have to be negative 10 over here and 10 plus 20, 30 over here, I think, for the second one, for it to reach one standard deviation. Yeah, the noise is very important information. Yeah. No one's disagreeing. But we usually want it to be small. We want it to be as small as possible, because if the noise is small, then if I'm inferring anything out of my mean, right? If I'm using mean in my t-test, if the HD is small, that's very helpful, because then I can trust my mean to some extent of my sample. So that is what Deviance by and his entire crew has been doing so far. It's been classifying, and it tells you that, hey, listen, you can trust a little bit here and there also. It's not that noisy, or dude, it's very, very noisy. That's what they've been doing. So it's assessing the mean. The mean does what? It gives you one figure. Like if I write M over here, it gives you one number, one hypothetical value. Sometimes it's like 2.6. You can't have 2.6 friends like we had looked at before, but it's giving you one hypothetical value. So these guys are coming up to then judge this guy, to judge the mean and like assessing it like, okay, looks good, good to go. So let's say that this over here is the population mean, right? Do you guys know the difference between population and sample? Can someone summarize? What is population? What is sample? Sample is a subset of the population. You usually take a sample from the population, right? So let's say, usually you will not know the population mean, but let's say, chalo, I know it. In this case, I am all knowing and I know it. Let's say the population mean is 50. Okay. Let's say I take a sample and it has a sample mean of 48. And I take another sample and that has a sample that has a mean of 36, which is closer to the population mean. 48, right? This guy is closer, this sample. And that's a problem that happens now, that in across samples, if I take more and more samples, right? Let's say I take more samples. Over here, if I take one more sample of M, this will give me, let's say, 46. If I take one more, it might give me, let's say, 26. So the question is, let's say if I take one sample, how do I trust that thing? What is the standard deviation doing? It's assessing the mean in terms of how noisy it is, but only for one sample, for that particular sample that you have taken. But what about the possibility of all these other samples? What about those other samples, other possible samples that could exist? That's where this guy comes in, standard error bot. His job is to assess the mean. He's also doing something similar, but if the sample was taken many times, like we did over here, right? I didn't just take one sample and look at the standard deviation of this one thing which assesses the mean. No. We took it multiple times, multiple, 3 over here, 4 over here. So that's his job, assessing the mean if the sample, okay? If we're not actually going and taking like, this is one sample, I've taken 100 over here, another 100, another 100 people. No, we're not actually sitting to do this 3, 4, 500 times. We're not doing that. So the standard error's job is to estimate this. That if I had done this, then how much is my mean differing from that? Is it differing by a lot or not? Or rather, like how close is it to the population mean? Again, we don't know the population mean. We'll never know it in most likelihood. But it's trying to do its job to estimate that. So very simply, it's saying that, listen, deviance by and variance kind of stuff. You have done a good job telling me about this one sample come in. You've told me that it deviates this much, and you've gone through the whole process of squaring it and giving me the HD and stuff. Awesome. But why should I believe you guys about this particular sample in the first place? I have no reason to believe you. You have collected some sample, for your dissertation or something, and some 30 sample they tell you to get and stuff. Okay, fine. But why do I believe that this sample is actually even anywhere close to the true population mean? I have no reason to believe you. Yeah, he's the critics board. So he's not impressed. Look at him. Look at his face. Look at his eyes. Look at his eyes. He's not impressed. He's like, okay, cool. I saw your standard deviation. It's small, but I am not impressed. So he's like, listen, there's 2 samples. Okay. 1, 2. Maybe you will see over here. Let's take an example over here. The first sample is telling me that the mean is 10. The second is telling me that the mean is 12. Who do I trust? They're both telling me that the standard deviation is 4. Standard deviation is not telling me anything about who to trust. Both are relatively small, so that's good, but it doesn't tell me who to trust. Now, the first sample has a sample size of 30, and the second has a sample of 300. So now who do I trust? Which one do I trust? Do I take 10 or do I take 12? Why second? Large sample, so you take 10. Why 10? Which do I trust? I got 2 different answers now, and I don't know what the population mean is. Maybe the population mean is 10, maybe it is 12. I don't know. So who do I trust? Larger sample, so more generalizable. Error could cancel out if sample is large. See, that is a good assumption to have, right? Like as your sample size keeps increasing, right? Let's say this is your data. Okay. And let's keep like this. One more. This is the population. This is the true population. This is a hypothetical drawing for now. So as your data keeps increasing, this distance, right, between the population mean, this is the mean, no? The population mean and your data come in gets smaller and smaller. That's a good assumption to have. But based on that only assumption, I cannot really say that this is the better one. I agree with your reasoning that the error could cancel out, but maybe this sample was so properly homogeneous and random sampling and everything, it was so representative of the sample that it could possibly be that this is also closer to that. This is closer to the population mean. I mean, look at the standard deviation; they're identical. So you know that one is not more noisy than the other. Both are equally noisy. So saying that one has more, cancels out the error than the other, we can't really make that claim so far. So which do I trust? First or second? No clue. See, that's the problem. Can't trust any. Don't know. When we started out, we're like, yeah, this guy seemed like a green flag, but clearly not. Can't trust anymore. So which do you believe? We don't know now. Which is why this guy comes in. He's here to do his job. His job is to listen and judge. So he's going to judge both of them equally. He's going to judge them properly. He's going to judge. That's his job. Look at his face. You know that's a judgmental face. He judged you the second you walked in. So he's going to judge between these two and, restore your trust a little bit. And how does he do that? He calls on the standard deviation. He tells him, like, this is the standard deviation, right? If I put this under a square root, this is your standard deviation. I'm not putting square because already 1 and 2 are already here. And he's like, listen, I'm going to put you on a trial. You are going to be judged. Why? Because what is the thing telling you? How noisy your data is. And he's like, I need to verify, cross check and everything to make sure that, I can trust. So he's here to do that. So what he does is he compares the standard deviation to the sample. He's saying, okay, I'm seeing that you are noisy. Okay. But how much noisy are you compared to your sample? Compared to your sample, how noisy are you? If you are very, very noisy compared to the amount of people in your group, then I can't trust your mean. Because what is this telling me about the mean? No. So if this is very, very noisy compared to the number of people in the group, whereas if it's less noisy compared to the number of people in the group, I can trust the mean a little bit better. Right? If you want a visual analogy, you can think of it like this. Say you are going to buy AirPods. Okay, you go to find, let's say, one AirPod over here. You have, okay, forgive my drawing, and another earphone you have. Okay, we're assuming that this one that you have picked is representative of all AirPods. So all AirPods. Similarly, this is, let's say, some other brand. So you want to compare, okay, how much noise is it allowing? Let's say the external world, you put it on in the showroom and you're seeing the noise. Right? Okay, wait, let me put that below it. How much noise is it allowing? Noise, despite you wearing it. And let's say you put it on many times, like say 10, 15 times you've tried it. And okay, every single time, wherever you went out on the road, traffic, wherever, it was fine. It wasn't very noisy. And over here too, how noisy is it compared to the like times of, you've gone outside and put it. So if this is like, let's say this was noisy only 2 times out of, you went there, it's, the noisiness over here. Then you can have a better faith in all airports. And if someone comes and asks you that, hey, is the airport good? You'll say yes. As opposed to the other one, which is, let's say, 7 times noisy. Now it's not 7 out of 10. Here I'm just giving an analogy. So you would have difficulty trusting this no-name brand. Again, this is just a visual analogy, not the math side of it. So you can see here, that's what it's trying to do. It's comparing the noisiness. And for that, it has to put the standard deviation on trial. So how much noise compared to the sample? And the way it does it, it's just taking an average again of the standard deviation. It's taking an average of the standard deviation. Almost poetic. You can't escape the average. Because all it's doing is it's this scatter. I can write like this. Standard deviation divided by n. That's all. Standard error is equal to standard deviation by n. On average, I think it's written somewhere over here. No. Like on average, it's seeing here that, okay, you're being this noisy, fine. But can I trust your mean? Now, something to note is that it doesn't only add n, it adds a square root over here. Any guesses why? Any guesses why it adds a square root? SD is under the square root. Okay, that is one reasoning, but there's a different reason. I'll tell you why. Because as the n keeps rising, right? As the n keeps rising, let's say I have a value of 2 over here. As the n keeps increasing, the total value will keep dropping dramatically. Let's say if this is 10 versus this is 100, the drop is going to be very huge. So the square root softens this. It's like a correction. It's like a very gentle, like, listen, don't go from, like, the knobs used to be there, the volume thing. You don't want it to go from like 0 to like 100. It's like, listen, let's correct it a little. Why though? Because as you keep increasing more and more, it keeps like dropping it more and more, which is like 2 to 3 times more and 10 times more. So you want it to compare properly. So we add this correction over here. It's like a scaling to smoothen it out a little. There is a technical reason. It's a scaling factor. That, listen, if it's dropping, don't make it drop too dramatically. Drop thoda aaram se. Okay, we can like do a comparison also. So basically his job is to tell you that, listen, your mean is not stable, dude. The mean that you calculated, right? You assessed it also with SD. I'm not certain about it yet. That's his main job, to tell you that we are uncertain. Okay, before I go into that, let's see over here what that looks like. Okay, let's compare and get a better idea. Okay, so we'll take over here. Let's say the SD is, we'll do a standard error over here. So what is it? 4 divided by 30. Tell me what value you get. Leave the square root for now. Here I'll add the second thing. 0.133 divided by 300. And for 300, it should be 0.0133. Yeah. Right. Now do it with the square root. Okay. Do it 4 divided by square root of 30 and 4 divided by square root of 300. What do you get? 0.74 and 0.2, what was it? 0.23, I think. 23. You see the difference? Over here, it moved an entire unit place. Whereas over here, it scaled it properly. It's comparable. So here I know that the standard error of the first sample is 0.74. Because look at this, it went down dramatically. One entire unit, it went down. Whereas over here, it's scaled. It's softer. It's gentler. So here you can see the standard error of the first one is 0.74, and standard error of the second one is 0.23. What does this mean? It means the smaller it is, it means I can trust my mean better, because this is less noisy now. This is more trustworthy compared to the first one. Both are good. It's not like the second one is bad either, but this is better. 0.74 is not that bad, but the second one is better. So if there is a true population mean, if it's 0.23, It's not that far apart. Whereas for the second one, with the population mean, the distance will be slightly higher if I keep taking a sample again and again and again. Right? So this is very simply the standard deviation of that sample of means. What does this mean? It means, let's say for the first one, right? What was my sample? 30. So say I took many samples, many samples of 30. It's saying that when I take this many times over here like this, the standard deviation of this will be 0.74. Whereas for the second, when I do it with 300 again and again and again, the standard deviation here will be 0.23, which is smaller. It's deviating smaller from the population mean, which is good. We don't want it to be very far from the population mean. Does it make sense? So it's such simple math. Standard deviation divided by square root of n. But it's doing something very cool. It's estimating this. Okay. All right. We'll move to the next thing. What is confidence interval? They're all connected, by the way, all of them, even confidence interval. They're all mila over, like, they're all involved with each other. This is the reason for my downfall. By confidence interval. Repeat what? See, the standard error part. It's basically just telling you that, listen, your mean, is it very stable or not? Like over here, you can see, right? We got the first sample, which had a smaller sample size. It had a bigger standard error compared to the second one, which had a smaller standard error. So the second one is more trustworthy when I'm talking about the population mean. Yes. What is the confidence interval, people? They say, what, 95% chance, something, whatever. This is the last thing we'll take for today, because standard error and confidence interval take some time to sink in. Any guesses? What is what is the confidence interval? I'll give you a hint. Standard error boy is a part of him. They are a part of the same team. They're colleagues. They work together. No one wants to try. Taken. They'll teach you something like it's the 95% chance of it'll give you a range, right? It'll be one range here, one range below. It'll be like, 95% chance it's within this range. This is wrong. They'll say something like 95% chance the true mean is there. That true mean or the population mean is within this range, which is wrong. Incorrect. That is not what the confidence interval does. What is it doing? It's very simple. It's telling you that, listen, if we repeatedly took random samples, right? Your population is there. You keep taking samples again and again and again, right? Let's say this is the population. You keep taking samples again and again and again and again and again. You keep taking randomly. How many times? I don't know. You just keep taking. If you keep taking repeatedly the random samples from the population and create the 95% confidence interval, which will show how to create, about 95% of those intervals will contain the true population mean, while 5% will not. So you see here, right? It's not about whether, how likely is it, like if there's a range, right? Let's say lower limit and upper limit. It's not about how likely it is in this or not, like, between this. It's whether it's there or not. That's it. It's about membership. Are you a part of this or not? Right? Over here, it's like if this, let's say I've taken one sample. Does this have the population mean? Yes. Does this have? Yes. Does this have? Yes. This have? No. This has? Yes. So that you can see here, population mean, if it's like something like 10 or something, you'll see that 95% of them have this gap, whereas 5% don't have it. And I'll show you what that looks like, what that means. Right? Let's say I have like 10 samples over here. Because I could not take 100, I'm just taking 10 for now. From them, 95%. Here it shows 9 because 90% ho jata hai, fine. Let's assume like this 5% over here, let's kuch. 95% of them have the true mean. These are all individual samples. Huh? Let's say I've taken a sample of like 100 here, 100 here. I've taken multiple samples. 100, 100. So this has population mean, this has. This guy does not have it. That's the bare idea of it. Like this should make it easier to understand this one diagram. Right? You can see all the green lines show what you can see. This middle dotted line, that's the population mean. Again, we don't really know it, we are estimating it. 1 second. Yeah, we don't really know it, we're trying to estimate it, but you can see here that this guy has it, this guy has it, this guy has it. You can see they're touching it. No, they're touching it, barely touching it. Has it. Everyone here has it. This guy does not have it. So 95% will contain the population mean. It's not 95% chance of it containing. No. 95% will contain. So as you keep taking more and more and more, 95% of them will have the true mean. But we're saying that 5% won't. We're giving that leeway that chalo, at least 5% won't have it. Why does that guy doesn't contain the population mean? Because of the way you might have collected data, no? Say you're studying depression in young women. Okay, let's say the true population mean is 25. Say you have collected one sample, you got 20 to 26 contains, check. You took one more sample, 24 to 28, this also contains it, check. See, you went to one more sample now. This sample has like, most of the protective factors. Let's say that they tend to come from wealthier families. They tend to come from more, more liberal, more open-minded families, which allow for a different good amount of things that don't pressurize for marriage and stuff. So their scores are slightly lower. So does this contain the true mean? No. So that's what we're saying, that we're allowing that leeway that some might not include. Yes, that 95% of them will contain the population mean. But again, we can't know this. The 95% is usually what we allow for it. It's not a fixed value in the sense that, that we have found it in nature. It's that we are allowing 95%. Because if it's 99%, the range, right, let's say lower and upper, tends to get bigger because there's more possibility now. It might be between these two very big numbers. Whereas if it's 90%, the range tends to get smaller. So a sweet spot is 95%. Not too big also that it's, it can falsely control it. Or what does it mean that we allow? We allow as in we are permitting it. I'll show you what that looks like in the formula also. We're intentionally setting the limit for 95. So this is what the formula looks like. And you're like, I have seen this guy before, because standard error, we just saw him. And what are we doing? We're very simply taking the mean. We're saying that, listen, add and subtract the mean with the standard error, which is multiplied at 95% confidence, or 95% of the area under the normal curve. So that is what we mean by we are allowing it, that we're controlling it. I'll come to it, but let's understand the formula from in the next few slides. I have it. Don't worry. So this is the formula for it. Mean plus or minus the z value plus standard error. So the z value ends up being usually 1.96 for 95%. If you look at your normal distribution curve, you'll see here, right? One standard deviation, 95%. No, sorry, 2, I think. It's slightly over 1. Or 2, almost 2, 1.96. No, that's the z value. No. Let's not go into significance. We'll touch p values later. Don't confuse this with p values for now. P values we'll touch later. Z is basically a transformation, right? That if I take my data, it transforms it such that the mean is 0 and standard deviation is 1. -1. I'll cover Z score some other day. But for now, you can see here that at 95%, it's 1.96. So you can basically take any data and turn it into a Z score. Okay. Because let's say does anyone know the formula of Z score? X minus M divided by standard deviation. So let's say that my X is, let's say my mean is 10 and my standard deviation is, let's say 2. So if I take X as, let's say 10 only, so 10 minus 10 divided by 2. So that's what? 0 divided by 2. How much is that? 0. Yeah. So the mean will always become 0. You see, it's forcing the mean to be 0. So let's say now that I, instead of taking X as 10, I'm taking X as 12. 12 minus 10 divided by 2. Kitna aata hai? 2 divided by 2 equals to 1. So this becomes my first standard deviation, right? Because the standard deviation is how much? 2. So it keeps pushing ahead and ahead based on the standard deviation. So let's say now I take 14 minus 10 divided by 2. That becomes 4 minus, 14. 4 divided by 2. So now second standard deviation is 2. So you're seeing it's transforming my data. It took all my data and now it's putting it in this form. Makes sense? I can keep going. If I take 16 now, it will put it as standard deviation of 3. And that is how our intelligence tests also work, right? 100 plus 15 is first standard deviation. Why? Because SD is 15 and this is mean SD. 100 plus 15 plus 15, again another SD, 130. That is second standard deviation. So that is the basic logic of Z score. So we are saying that 95% of people will be within this range. That is from 1.96, negative 1.96 to positive 1.96. Usually it's that. So we take 1.96 generally, which is why I have put 1.96 over here. So coming back to that question, someone asked me that the 1.96, the plus or minus, okay, plus or minus because below and above, right? Plus or minus sets the width of the interval, right? The interval that we have, it sets the width of it, how big or small it is, based on how much uncertainty we are willing to accept. So if I'm taking 99% confidence, we'd use this value instead, not 1.96, which gives me a wider interval because it can because I want to see within 99% confidence, right? It's like you're going fishing and how big the rope is. If I'm taking 99% confidence, I'm taking a bigger rope because let's say the population mean is here, my 99% will catch it. Right? This is 95%, 90%. The width gets smaller and smaller. So this guy caught it. But what is the problem with that? Now my rope is much bigger. It's too big. Okay, the 95% missed it, but it's fair enough. It's not that far. So it'll likely catch it. The 90 is not even close. This is the population mean, which is why you can see here. How do we know? We take this as a rule, right? We're taking 95% as a rule. That is how much uncertainty we're allowing. We're allowing 5% uncertainty. That is what we're doing. We're allowing it. The second you pick this, the second you pick 99% or 95%, you are allowing it. You are controlling it. Because if I was saying 99%, I would be coming up till here. What was it? 2.576, no? 2.576. So let's demonstrate what this looks like for our data, right? That we had taken before we had calculated the SE and stuff. So for first sample, right? Let's say we have a mean of 10, this and our standard error. What was our standard error? 0.74. So let's calculate quickly the confidence interval. So 10 plus, okay, calculate this first, 1.96 into SE because it's a multiplication. Then add and subtract. Done. What is the positive range? I'll put it as a this over here inside the bracket and separate it by comma. Can someone cross check? Yeah, 11.45 and 8.54. No? Did you take 0.74? Yes, negative. No, not negative 11.54. The range doesn't have to be negative because here it tells me if my mean is what? What is my mean? 10. The true mean, it will lie here between 8.54 and 11.45. So 8.54 or 55, whatever, and 11.45. This is my mean. So it's giving me that range now. The interval, right? The one of the intervals before I showed you here over here. These all are intervals, right? We just calculated one interval such. So it gave me one interval for my sample, which is 8.54 and 11.45. Let's for curiosity's sake, see how big it is. 11.45 minus 8.54. See how big this is. And tell me how big it is. 8.54. 2.91. Okay, let's just keep this in our mind for now. We'll come back to it. Okay. For the second one, 12 plus or minus 1.96, again, bracket here, and our standard error was 0.23. What is the range you're getting? Can someone cross check? Because if the mean is 12, how is the range not including the mean? Okay, I'll write 11.55 and 12.45. Okay, now coming back over here, calculate the difference again. 12.45 minus 11.55. You see the difference? 0.91. You see this? It's a smaller interval than this. This is much bigger as an interval at 95%. Both are at 95%. Whereas this is smaller. So what does the confidence interval do? Right? Because the sample of this was small, the interval is huge at 95%. Because the sample was big, the confidence interval was small. So when we're getting a smaller confidence interval, it's giving me hope, a little hope. It's being a little nice. It's saying that, listen, you're less uncertain. Okay, I'm not saying I'm certain. No. I'm saying I'm less uncertain than this, than the first sample. So when your confidence interval, when you're repeatedly getting it smaller and smaller in different places, you can be less uncertain. And that is the job of the confidence interval. So you can see here that the first sample's CI is wider, and that reflects more uncertainty because it's based on fewer data points. Whereas the second one, both at 95%. Despite that, the second one is narrower, which means that we have greater precision in estimating the population mean because of the larger sample size. And this is important because usually, let's say I'm doing a t-test, right? Let's say this is my first mean, this is my second mean. Usually, it's a good sign if the CI of this and the CI of this do not overlap. Let's say this mean is 10, this is, let's say, 12. Again, same thing. Right? We'll take the same only example. If I'm comparing the two means, what was it? 11.45 and 8.454 and 11.45. So if I draw it here, if this is 10, this is 11. Let's say this is 11.45. Okay, this is 12. And what was it? 8.12 का क्या था? 11.55 and 12.45. So it's here 11.55 and 12.45. So usually they ask this in NET exam once they asked it. So you can see here the overlap is not there. This is the first. This is the second. No, I accidentally made it overlap, but it's really not overlapping. There's a difference. So that's usually a good sign. You don't want them to overlap. So that is the crux of confidence intervals. Any questions? Any doubts? Yeah. You can stop recording. You're anyways done. Just give me 1 second.
Can I stop the recording now?
Yes, you can stop it. No worries. Thank you, Sarah. To answer the question of why don't we want it to overlap. So in t-test, we don't want the two, we want to check if the two means are different, right? Oh, it stopped sharing. We want to check if the two means are different, right? Here we have two means. Here you're just comparing, right? These two, is it the same or is it different? So if the confidence interval of the first one and the second one don't overlap, that's usually a good sign. That's usually a good sign to show that they are different. They really are different. Or if you want me to draw it as a not equal sign. But we'll get to t-test later. But yeah, in that context, I think that asked once about the overlapping thing. Okay, what is the other question? What we use confidence interval for? This is the use. This is the use of confidence interval. The reason we are mentioning it in our papers is to show how wide this range is or how narrow it is. If it's narrow, we can then say that, okay, we have some amount of certainty. If it's wide, then it's a little difficult to trust whatever the conclusions are from that paper. We don't want it to overlap, because you can see here, right? From the first data, the mean is here, the mean is here. We don't want these to overlap. If the CI, the confidence interval, what does the confidence interval tell you? That the population mean is here. So if the population mean is here or over here, it might mean that it's closer and closer to the second mean, which means the difference might not be real. How are we linking wider CI or narrower? Because we are getting to know about wide CI and narrow CI because of the N, no? Because of the sample. And it's not because of the N per se, like with N, but it's telling us that at this confidence, the range of your mean is wide or it's narrow. It severely restricts what you can say with it. It's very difficult to interpret it, which is why you might wonder, like, why even bother with this? So it's just a measure of certainty or uncertainty. So when we write that, oh, there's a 95% chance it lies within this, no, that's wrong. Interpreting it gets much harder. If they don't overlap, there's a good chance that our samples might have different population means. It's a good sign. It doesn't prove that the samples are different. Okay, it doesn't prove it, but it's better. If they overlap, there's a better chance that they are not different. But again, it proves nothing. It's just a good indicator, which is why we end up using the t-test or something. But it's a good indicator. Again, you can't see it in that absolute term, because the confidence intervals whole job is about certainty or uncertainty. So if my t-test is showing a significant difference and the confidence intervals don't overlap, then I can have slightly more certainty. I can't say for sure they are different, but I can be slightly more certain that they are different. But if my t-test is showing significant, but my confidence intervals is overlapping, then my certainty drops a little bit. Not much, but little bit. If the study has good power and all, will touch power. But if the study has good power, good sample size, it is still, I can still believe it, but it still drops my certainty slightly a little. So it's all about believing. No. That's the thing, right? We can't claim anything. We're being very gentle over here with this. We're just saying that it's informing us on what degree I can believe this. It's soft evidence, not evidence really. It doesn't prove anything, but it just, to some extent, I can be slightly more certain about this, or I can continue feeling uncertain about it. It's not a direct claim. Even the t-test is not a direct claim. But okay, it's a better claim than CI. If their population means are different, then how is a good sign? It's a good sign because that's what you're doing with the t-test, right? You're trying to check, is there a difference between these two groups? But again, if you're not familiar with t-test, no need to worry. We'll touch the t-test later. Anyways, we'll be covering it. Okay, is that clear? Not going into covariance today. Any doubts? Any questions? Did it make sense so far? Because a good chunk, can we not have stats and biopsych on same days? What do you want instead? What is not heavy then? That's good. Light, and we have been trying that, but the thing is, all the subjects are pretty heavy, like research, stats, personality, biopsych. And the thing is, I can't put stats and biopsych, personality together, because then I'm taking 2 lectures a day, because personality would be slightly lighter. But yeah, I think anyways, biopsych will be paired with something like abnormal, which is abnormal slightly more fun, which you might be aware of. Yeah, I'm glad. So I hope so far it's clear. If it's not, you can go and rewatch, or you can make sense of it by watching some other things, ask for a resource, whatever it is, because this is the fundamental. This is the basics, right? Mean, standard deviation, standard error, confidence intervals, and variance. If you understand these, I promise you, you will make sense of everything else. You'll make sense of correlations, covariance, regression. You'll make sense of ANOVA. You'll make sense of t-test. You'll make sense of ANOVA. You'll make sense of everything, because it's all built on this. I am not even kidding. It's all built on it. So the non-parametric tests are not directly built on these because they don't really have a mean, right? What is the main logic? X, the raw score minus the mean. But they are not built on it. But you will still see traces of the same logic being used. A similar logic is still being used over there. So when I don't think in the foundational series we are touching non-parametric math, but when you look at Man-Whitney U or Kruskel-Walliska H, right? Yeah. When you look at them, they're using very similar logic, because this is fundamental. This is fundamental, comparing the mean with everyone else in the group. Yes. Okay. Has it made sense so far that we have been here? This is the third class of stats. The stats feel scary. Does it make sense now? Is it starting to sink in? Anything I can do differently to make this more digestible? Okay. Because see, I don't expect you to get it in like an hour. But if I explain the basics of it, the foundation, the way it works, the logic of it, it'll sink in. And then it makes sense eventually. Okay. Awesome. Today I'm not taking the MPhil prep thing because honestly I'm too tired. So we'll take that tomorrow evening. Okay, because I don't have a lecture tomorrow, so I can prepare for this, the MCQs. Okay. Awesome. I will see you guys tomorrow then. Can we have like dummy data and make sense of? Yeah, of course. We have revised towards the end. Yes. Which book can we refer to revising these concepts? If you want to understand the concepts better, go to Andy Field. If you want to look at solving it and stuff, the A-kissing and stuff works better. But the logic of it makes sense better in like Andy Field, if you have the time for it. It has a good chunk on standard error that you can go and read. It explains it pretty well. Yeah, no, we're not doing it today. I'm too tired. Been a very long day. But tomorrow I don't have a lecture, so at least I can do the MCQs. Andy Field's discovering statistics using IBM, SPSS, or discovering statistics using R. Okay. Awesome. Okay, bye people. See you.